Karissa received her BA in physics at Columbia University and her PhD in astrophysical, planetary, and atmospheric sciences at the University of Colorado at Boulder. So she started out like way out there in the universe, and now she's come home in here into the inner universe of biological beings. I love that. She had a postdoctoral fellowship in applied computational physics at Los Alamos National Laboratory, was awarded the Presidential Early Career Award for Scientists and Engineers, and was elected a Fellow of the American Physical Society. She described her work with epigenetics and came out as transgender in a 2018 TED Talk titled The Biology of Gender, From DNA to the Brain, which has over 2.3 million views. I encourage you to check it out. She is on the board of Equality New Mexico and is an advocate for LGBTQ people in the sciences. So, Dr. Karissa Sanbonmatsu, welcome.
Well, thank you for that wonderful introduction. It's such a treat to be here at Science on Tap. And today I'm going to talk to you about some new research we're doing in the biological sciences. And then toward the end of the talk, I'll just touch on some of the biomedical applications that we may see in the future from this line of research. Okay, so I want to tell you about sort of a revolution that's been brewing in the biological sciences for about the past 20 years. And that revolution is about sort of which molecules control our cells in our body. And so traditionally speaking, people used to think that it was all about DNA and proteins. And nowadays, I'm going to tell you that it may be RNA that's really playing one of the key roles. And RNA is sort of a molecular cousin of DNA. And some people call these RNAs the dark matter of the genome. And I'll explain why they call it the dark matter during my talk. And so inside our cells, we have many cells in our body, and each one of those cells is like a tiny city that's teeming with all kinds of little molecular machines doing all kinds of things. And these molecular machines are mostly made out of protein molecules. So you may think of protein as things that are in steak and chicken that you eat, but it turns out that almost all the things that do stuff in our body are protein molecules. And then these proteins can sort of carry cargo from one place to another. They can make the scaffolding of the cell, and they can do all the biochemistry in the cell. So it's mostly proteins that do the actual work of the cell. And so this brings us to what's called the central dogma of molecular biology. And this was put forth by, among others, Watson and Crick in over 50 years ago. And they wanted to sort of say how the information flows in a biological cell. And the idea is that the information starts in your DNA, and the information in your DNA encodes those proteins that we saw in that animation that are doing the work of the cell, most of the things in the cell, and making up the structures of the cell. So those proteins that make up the structures, the information encoding those is inside your DNA. And then there's sort of, in the old view of biology, it was these RNA molecules that were just these transient intermediates, and they'd pass information from the DNA to the protein. So here in this diagram on the left-hand side, we have the DNA, and then the information goes to this, what was used to be thought as just a transient sort of copy molecule called RNA, and then from there, it was manufactured by the ribosome into proteins. The ribosome is sort of this large molecular factory that reads RNA, and then it creates proteins. And so there are different processes involved in information flow in the cell. And what I just described to you in the central dogma is where you have RNA production from the DNA. This is called transcription. And then you have protein production from the RNA, and this is called protein translation. And then also another place for information flow is the DNA is also copied into other copies of itself for cells to duplicate and so forth. So this is the central dogma of molecular biology from the late part of last century. And in terms of the DNA part of this. So the information goes from DNA to RNA and then from RNA to proteins. So for the DNA, this is composed of a large sort of ticker tape. It's a polymer molecule, or it's a very long string-like or spaghetti-like molecule, and it's made of these bases or nucleotides. There can be 4, adenine, guanine, cytosine, and thymine, or A, G, C, and T. And then there's sort of a repeating sequence of these that is strung out, and there's about 3 billion of these side chains or bases in each of your cells that make up what we call the human genome. And this DNA is wound up in a double helix, and then that double helix itself is wound up into all kinds of other formations. So the first formation it makes is it wraps around these proteins that are sort of like molecular spools, and this is called a nucleosome. And then those nucleosomes then kind of compact into this big kind of globular mess that is a chromosome, and then there are 23 chromosomes in the cell, and this is the structure of your chromosome. Okay, so it turns out that the structure of the chromosome is very important to understand, because this can actually control how genes are turned on and off. Actually, I may go back one. I skipped a point I wanted to make. And that the idea here is that the genes are basically segments of this DNA that we're showing. So this DNA can be divided into thousands of segments, and each segment is basically a gene. And that gene encodes for a protein. And so when the region corresponding to that gene is wrapped around and highly compact, in that case, the gene may be silenced or downregulated or essentially shut off. And then when it's more expansive in that case, then it will be activated or turned on. Okay, so then we can talk about what we're doing in Los Alamos to look at the structure of these chromosomes. So here we're using supercomputers to try to understand how the DNA actually is folded around those spools and how those conglomerates of spools can compact or expand. And so recently we did a big calculation on the Trinity supercomputer, where in this case what we had was about 400 of these spools and the DNA wrapped around them, and then we simulated all the atoms in this whole complex on the supercomputer to try to understand how it sort of undulates and breathes and compacts or expands. And in total it was about 1 billion atoms, so it was a quite large calculation. And it turns out that to this day no one's actually seen a chromosome at atomic resolution, but what we can try to do is simulate what we think they should look like using supercomputers, and we use a lot of experimental data to build these models. So this was the DNA part of this central dogma. And then to continue on this, it turns out that when the baby is developing in the womb, all kinds of decisions have to be made. And the shape of the DNA plays a huge role in making these decisions. So in this case, whoops. Okay, so let me skip this slide. Okay, so in this case, the idea is that if all the cells in your body have nearly identical DNA sequences, then how can they be different from each other? So in our body, we have muscle cells, we have brain cells, we have blood cells, we have liver cells, and the idea is that in these cells are completely different from each other, but they have nearly identical sequences. And so if that's true, then how can this be? And the answer is that the genes are turned on and off differently as the egg is fertilized and dividing into 2 cells and 4 and so on. These different decisions are made in terms of turning genes on and off to convert one cell into a liver cell or a muscle cell and so forth. And it turns out that the shape of the DNA is absolutely critical in controlling that. And so I'll flip back to this slide, and so it turns out that in controlling the shape of the DNA, the RNAs play a huge role in controlling how the DNA is shaped and therefore turning on and off the genes. And it turns out that one of the big surprises that came out as people were learning more about the chromosomes and the genome is that most of the genome actually doesn't code for proteins. Originally it was thought that the main purpose of the DNA was to have the information to make the proteins, as we learned in the central dogma in all the textbooks, but it turns out that actually only 2% of the genome codes for these proteins that do things in the body. So what does the other 98% do? Well, 90% of it is called the non-coding portion of the genome, and we also call this the dark matter of the genome, and that is because it's sort of this area that's not really as well understood as the regions that code for proteins. And so basically the central dogma has been revised in the last 10 or 20 years or so, and that it turns out that RNA molecules are playing a big role in regulating how the DNA is turned on and off. And so the 90% of the genome that encodes RNA and these RNAs that do not code for protein, we call those non-coding RNA, or the dark matter of the genome. And so these RNAs play different roles. So we have the RNA can act back on the DNA, controlling the fold of the DNA, and also the RNAs can also regulate if the proteins are manufactured or not. So the RNAs are doing a lot more than just, in the old view of things, being a transient template to transfer information from DNA to RNA and then RNA to protein. The RNA itself is actually folding up into these machine-like things and actually doing a lot of interesting things in the cell. And so, so what are these non-coding RNAs? Well, nowadays there's sort of two classes of the RNAs. There's the short ones and then the long ones. And so in the 90s and 2000s, the short RNAs kind of ruled the day, and these were big discoveries. In the early 2000s, Craig Mello and Fire discovered these short interfering RNAs, which are also related to microRNAs, that these have the effect of basically finding one of those transient messenger RNAs and chopping it and shutting off the central dogma at that point. And then just a few weeks ago, Jennifer Doudna won the Nobel Prize in Chemistry for Crispr, which was this discovery on how RNA is used to target regions in the genome and cut the genome. And then it's been used in biotechnology to actually do gene editing, where you can sort of alter genes at will almost. And then in terms, and what we're showing here on the left are these structures of some of these RNAs. So on the bottom is this shorter RNA that's involved in RNA interference. It's a double helical RNA. And then in the Crispr complex, there's a smaller RNA in red here, and then it's bound by all these proteins. And that whole complex gloms onto DNA and facilitates the editing of the DNA. And then in terms of long non-coding RNAs, this is a whole new class of RNAs that really just came up in the last 10 years or so. And in this case, there are these great big RNAs, and people know they're important. Like if they knock out one of these big RNAs, it may change the development of cancer, or it may block development of a certain tissue type. It may alter brain function, may do lots of kinds of things, but no one knows what they look like. So because they're so long, I mean, these are tiny, they're inside your cells, but in terms of the field of molecular biology, they're quite big and unwielding and very hard to work with. So there's almost no structural 3D data out there for any of these thousands of long non-coding RNAs that have been discovered. And so one of the studies we've been working on here in Los Alamos is on a molecule that we call Brave Heart. And this is a collaboration with our colleagues at MIT, Laurie Boyer, and a new collaboration with my colleague at Lethbridge University in Canada, Tushar Patel. And a lot of the work on this project was done by Scott Hennelly, who's shown up there, and then also Dunam Kim as well. And so this RNA system, it's one of these long non-coding RNAs that Laurie and MIT showed it played a really important role in the development of the embryo. She happened to be working with mice, and she showed that if you remove that RNA, then you no longer can have heart development. It's a sort of, they call it a master regulator. It's at one of the key decision points to make the heart in the mouse embryo. And she came to us, and she wanted to know, what does this RNA look like? So we went into the lab, and we started doing some biochemistry experiments. And we used a technique called chemical probing, where in this case, we essentially make up the RNA. We add different chemicals to the RNA that only stick to pieces of the RNA that are wildly fluctuating or jiggling around. but they don't stick to the ones that are rock solid and don't jiggle around. And in that way, we're able to map out sort of a first step or a 2D map in 2 dimensions, like a flat view of how the RNA is organized. And it turns out that RNA is mostly organized in double helices, just like DNA, but there's lots of different double helices, and they can bifurcate into 2. Those can bifurcate into 2. They can all fold up and do a lot more complicated things than just plain old DNA. And so in the 2D diagram, we're looking at the different double helices, like helix 1, helix 2, helix 3. These bifurcate into 4 more, and so forth. And this is sort of the 2D diagram, almost a blueprint of how the RNA is organized. And then, in addition, Lori at her lab in MIT, when she started working on this, the people on the floor above her had there were other independent discoverers of the CRISPR technique, and so she applied the CRISPR gene editing to her cells, and she has this nice assay. They call it embryoid body beating. And so they have kind of heart cells that are beating on a plate. And then what she was able to do is show that when you knock out a very small portion of the RNA, that it actually destroys the beating activity. And that's shown in this little chart here, where we're looking at the normal wild type version. Then if you knock out just a small piece of the RNA, then it destroys the beating. And I should flip back and say that one of the main things that came out of our study of the 2D map of this is we found that we keep finding these, we call them right-hand turns, but these big, long loops that cause the helix to turn over. And so we thought that loop was important, so she deleted that loop using CRISPR, and she found that specific loop kills the activity of the whole RNA, and therefore development of the heart. And then also she did a number of follow-up studies looking at these, what they call cardiac transcription factors, or sort of gene expression levels important for the development of the heart, essentially. And she saw that if we delete that one loop area of the RNA structure, it knocks down the activity. Okay, so then the last thing we did is Dunam Kim embarked on this 3D study. So we're just taking the first steps into trying to get a 3D look at this. So the first aspect of the problem was to get the 2D organization of how the RNA is laid out, how many helices does it have, and so forth. But then the next step was, can we see those helices in 3 dimensions? And so the first step here was to use sort of a very low resolution method called X-ray scattering, or small angle X-ray scattering. And so the samples were sent to a X-ray source, and then we collected the scattering data. And then after a lot of computer processing, we were able to come up with these models of the 3D structure. And so we think that it's quite flexible, and it doesn't have one static structure, but it kind of oscillates between a lot of different structures. And so here, if you look carefully, sort of, for example, in blue and green, you can see these double helical pieces of the RNA, and then the other kind of more spaghetti-like features are the connecting regions. And then maybe I'll just flip back a second. So again, you can see the 2D map where we have the different helices. H1 is helix 1, H2, H3. Those are the different helices. And then you have these thin looping regions in between. And then when we try to get an idea about what it looks like in 3D, this is what we come up with. So this is consistent with our 2D map and consistent with the 3D scattering data that was taken. And then lastly, this is the final slide, but this is showing, comparing our 3D models with the actual experimental data. And from the scattering, you can make these envelopes that are shown here in the hashed blue. And then our model, what we did for with this yellow is we basically averaged the different conformations that we saw and weighted them by population. And then we do that, our density from the model is very close to the experimental density. And so all in all, this is one of the first ever 3D structures of one of these long RNAs that's critical in developing hearts. And in terms of biomedical applications, there are many. There's a lot of big drug companies that have big departments moving into the long RNA field with lots of money, so it's a lot more competition. But they're trying to find ways to make these RNAs druggable. If there are problems in heart development, could you use a drug to help with that? Other people are interested in growing artificial hearts, and since this RNA is key to the development of heart, it could be important for growing artificial hearts and also in regenerative medicine in terms of heart disease. So all in all, what we're seeing here is this revolution of RNA biology it's spreading out quite a bit, and we're seeing that big classes of RNAs are coming into the limelight, and there seem to be big players in how the cell works. So in terms of all the molecules in the cell, who controls the molecules? The RNAs are actually important players in this, and so the central dogma of the information going from DNA to this just template RNA that then makes a protein, it's being rethought now, and the RNAs actually do a lot of different things. Right.
A lot to think about there. One question that came to mind for me, and I want to remind everyone, you've got to write in with your questions, okay? And I'm sure you have a bunch of them from hearing Karissa's introduction to us. I began to think about, so what's the intersection between, or what's the interplay with a cell that is called a stem cell and seems to have every possibility, and what the RNA is doing in a stem cell that might be different from what the RNA is doing in a liver cell?
Yeah, this is a really great question, and it turns out that the stem cells are sort of like when I was showing about the embryo growing inside the womb, and initially you have these, the egg is fertilized, that divides into two, and those divide into four. Those are sort of kind of blank slate cells where the decisions really haven't been made yet to turn into a muscle cell or a blood cell, and those are very analogous to stem cells. Stem cell is kind of the same thing, where it's kind of a blank slate cell where you can trigger it to go into one of these cells. And right now it's still, I'm not really in the field, but from what I can tell, it's more art than science, where they have these different recipes to transform a cell into one kind of cell or another, but they don't quite understand what's happening at the molecular level. And we know the RNAs are important in these processes, but it's so early on, we really don't know what's going on with the RNA. So it has a lot of open avenues for research now.
Great. Let's see. I just want to make sure we kind of move from one subject to the next in a cogent way. So since we were talking about stem cells and how they might, how that interplay is, Chelsea asks, could this research be used to help cardiac muscle regenerate after a heart attack, a myocardial infarction, and if so, how?
Yeah, so I think in the long term, we'd like to use this kind of research to help with these regeneration processes, but we're really just like, this is one of the first times the RNA has been imaged, and there are many other RNAs happening, and then we have to see what all the proteins are that bind to that, and then map out all the pathways. So we're kind of a long way off from real practical applications, but for sure in the long term, we hope that we'd see a nice application like that.
Yeah, but you got to start somewhere.
That's right, yeah.
And I was excited to learn that your work on this is similar to what I've learned your work is with the ribosome as well, where you take actual imagery of components through like the cryo-EM,
Right.
and you've got your real physical imaging data from a physical thing, and then you're working it into a model so that you can build it out and put all the atoms where they seem to go and see how they come together.
Absolutely.
Yeah. That's just Amazing. And so I think also a good reason why you're working with so many collaborators.
That's right,
Because
yeah.
everybody has a specialty.
That's right. It's very interdisciplinary, so we need expertise from all kinds of areas of science.
Yeah, yeah. And that's a really good message for people who want to get into research going forward. It takes so many different kinds of specialties to move something along.
That's right, absolutely.
Yeah, and so many. The more we learn, the more we realize we don't know about something.
That's so true. Yeah, like a lot of these projects have people in cell biology, biochemistry, computer science, physics, material science, all kinds of areas, and we try to bring everyone together and get everyone speaking a common language to solve the problem.
Here's a question, and I think a bunch of people are interested in this right now. Colleen asks, how does our RNA differ from a virus's RNA, and how they use it to reproduce?
That's a great question. So it turns out that it depends on the kind of virus. So some viruses have DNA inside of them, and some have RNA inside of them. And just like we have a genome where our proteins are encoded in our genome or our DNA, the virus also has a little genome. It's very tiny, maybe not very many genes at all, but it has a genome that encodes its proteins. And so some of those viruses, for example, like the COVID-19 virus, has a genome that's made out of RNA, actually, instead of DNA. And then these viruses are very sneaky. They get inside your cells, and they trick your cells to using their genome and make copies of their proteins instead of our own proteins. And so understanding and imaging RNA is really critical to understanding how something like COVID-19 works as well. And also a related topic is the Moderna vaccine that's being worked on now. This is an RNA-based vaccine. And most vaccines are protein-based in terms of antibodies. But the Moderna one is one of the first ever vaccines where they're using actually pieces of RNA that are inside a little capsule. And the idea is this gets delivered to lung tissue, and then the RNA comes out, and then it then goes into the ribosome and makes proteins in the right place that somehow help in terms of the vaccine. So.
Oh, fascinating. Ben wonders, do I understand this study is about coding proteins that become heart muscle? Is just about coding proteins that become heart muscle? Not, is it? Or is
okay,
it?
So what we're looking at are kind of, in terms of how the heart develops, in the end there will be proteins involved in heart muscle, for example, that are important. But before that happens, in the embryo, the cell has to make the decision to become a muscle cell or like a blood cell or something like that. And what we're trying to look at is the control mechanism on that decision-making process. And that has to do with how the chromosomes are folded up, like I talked about in the beginning. And that has to do with the RNAs. So we think the RNAs are controlling somehow how the chromosomes are folded up, and that in turn can tilt the decision toward a certain kind of muscle and so forth.
Mm-hmm, mm-hmm. Is all cardiac muscle the same, or are
no,
there...
there are different kinds of cardiac muscle. Yeah, so that's another good point. Like for each tissue, for example, in your brain, there are all kinds of different brain cells, and so sort of a big decision has to be made if it's going to be a brain cell or a muscle cell. But then after that decision, a lot of other decisions get made on what kind of neurons the cell turns into and so forth.
Yeah, and where they all are with respect to each other.
That's
and
right.
how they all
it's
and how they all
mind-boggling.
come online.
Yeah,
in
absolutely.
what sequence.
That's right. Yeah. It's amazing that we can be alive and talk to each other.
It's a good thing we've had about 3.5 billion years for
right, exactly.
the experiment to run.
That's right.
So that it worked out. Here's an interesting question, and this is from David Groh. Very nice presentation. You mentioned CRISPR. As our understanding of related areas increases, and improves, I see tools such as CRISPR reshaping our world. What are some of your thoughts on the ethical landscape we may be faced with?
Yeah, I think this is so important. And Jennifer Doudna, when she made the discovery a while ago, but then shortly after, she and others anticipated that there would be a lot of ethical issues. So they have set up a lot of bioethics studies and committees and so forth. And there is several moratoriums worldwide on how you actually use the gene editing. So I think with any big breakthrough in science, you have to be careful. It's a tremendous responsibility on how it should be used.
Right.
So right now in the scientific world, people are mostly using this in animal studies and just plated cells, cell cultures, and so forth, mostly for research purposes right now.
Mm-hmm, mm-hmm, mm-hmm. This would sort of be a good almost ending question, but I'm going to ask it right now. Why was the heart RNA selected for study, and which other organs' RNA might be next in line for research?
That's a great question. So we happen to be one of the first groups doing the structural studies of the RNAs, and we started in about 2011. And I went to a conference in Cold Spring Harbor, and at that conference, two people approached me. One from the UK who was a plant biologist, and so we have another project on RNAs in plants that control how the plants flower in the springtime. And then the other person was a postdoc at MIT, and her boss was Lori, and they did the Braveheart project. So it was just kind of happenstance. They were looking for people to collaborate with, and we were excited to work with them. They didn't know how to do structure, but we didn't know how to do CRISPR or the cell biology, so we joined forces. And Lori's group are experts in heart development and so forth. So they're a pretty high-powered group in heart. So that's how that happened. And there are so many. The RNAs play important roles in all of our organs. And me personally, I'm most interested in the neuroscience applications, because in brain function, a lot of brain disorders, Alzheimer's, autism, Rett syndrome, the list goes on. Just how memory works as well. The RNAs and also how the chromosomes fold play important roles in those. So I'm keen on that. But also we do have a new project at Los Alamos where we're working on how coronavirus affects the shape of our chromosomes and how RNA can be, how it plays a role in there as well.
So coronavirus affecting our genetic material.
Yeah. So it's more this Los Alamos project is more focused on the first part of my talk on the chromosome shape, and so we're trying to see how infected people's chromosomes differ from non-infected people, and that there are a lot of subtle questions in coronavirus now that people are kind of clueless about, like why are some people more susceptible than others? Why are older people more susceptible than younger people? How come your blood type O makes you more resilient than other blood types? People, it's so mysterious now. There's all kinds of questions. Like the focus in research has been on the virus and what does it look like in the antibodies, but there's a lot more big questions out there in terms of long-term effects and so forth, I think.
So another kind of traditional thought that we've had for over 100 years is what drives evolution is natural selection.
Yeah, right.
But it seems to me that there could be, once we had them extant, if they can be extant on Earth, viruses, maybe there's a viral aspect that drives evolution as well. Is what you're talking about with how it might affect our chromosomes.
Sure, and I think the biggest example is a retrovirus, where for example, in HIV, the genome is made out of RNA, just like COVID, but it's called a retrovirus because the RNA actually winds its actual code into your DNA. It's like backwards of the central dogma, the information.
Yeah.
And then that can be passed down to your generations.
Right.
And then in terms of evolution, in terms of these long RNAs, it's so early in the field, people are still kind of making hypotheses, but some people think that these long RNAs, they evolve so rapidly. They evolve very, very rapidly. Some people think they're kind of evolutionary, like the advanced team, exploring different directions in evolution space to see what's a beneficial function and so forth. So that's a really hot topic right now.
Yeah, yeah. Fantastic. Happy to get any more questions out there, folks. Keep asking. Let's see. Why haven't, before this time, people imaged the structures of many long RNAs?
Yeah, that's a great question. So it turns out that there are two reasons. So RNA itself is kind of a very finicky molecule to work with. It actually, if you just leave it there sitting, it actually cuts itself. And then there's all kinds of things in your cells that are made to cut RNA. And the reason for that is because a lot of these viruses do have RNA genomes. So you have a lot of things, they call them RNAs, it's an enzyme that cuts RNA.
Right.
And they're flying around your body, they're on countertops, like in the lab, they're everywhere. And so they can really cut apart your RNA really easily. And that's one reason. Another reason is it's they tend to be a little bit more floppy. So it's hard to really get them to sit still and take a picture. They're always like fiddling around, you know. So for that reason, that makes it tough as well. So it's much harder to image these kinds of molecules.
And then I don't know when did the technology actually catch up to be able to do the imaging?
That's a great idea. So some of the technology that's used today, one actually is sequencing. This high throughput sequencing helps us make these 2D maps. And in RNA, we have to kind of have that 2D map because it's so hard if you just have a jumbled mess of noisy microscope data to see which position, 753 goes here, 752 goes here. It's almost impossible. So if you have that 2D map, that gives a nice framework. And then next, where we're going with this next is we have a lot of things. We're working right now on cryo-electron microscopy of these molecules and other RNA molecules. And so this has been another breakthrough in science where the direct detector solid-state cameras were used to do imaging. And so in this case, they freeze it, and then they have these expensive microscopes, and they have these expensive cameras, and they're able to go down to almost atomic resolution. So that's another piece of technology that's been helping us.
And then to say nothing of the compute power that's
required
to make it all make sense.
The compute power helps a lot with the modeling, and so we are using a lot of the high-performance computing platforms at Los Alamos, and also not only with the modeling, but for the cryo-electron microscopy, there's a lot of image processing. They have terabytes of data, and they have to churn through it. So the compute is a big bottleneck in using a microscope nowadays. So.
I've heard about the catalog that we have of the various sequences of HIV. Are we going to be building out a catalog of RNA sequences?
Yeah, this would be a great thing to do. There's a few projects, efforts. There's one in Japan and a few others working on this, but they're not quite. It's so new. It's not quite so systematic just yet. But it's something an interesting direction.
Yeah. Let's see. Ooh, I think we've got another question. Yeah, I've got a mouse down some way. Where's my pointer? Given that some viral RNA, maybe she meant to say DNA. I trust her. She knows what she's talking about. Sorry. Technical issue. I can't. Hey, Mel, can you give me an assist here? I'm trying to read the question from Chelsea that came in at 6:18. Can you read it?
I can read it from here. You want me to read it?
Yeah.
Oh, yeah, actually, something. Well, here we go. I got it. Thank you. Given that some viral DNA remains in our genome throughout generations. Have any links been made between this and your research, or any impact this may cause?
That's a great question. So we talked a minute ago about the retroviruses, and then
Right.
the viral DNA gets into your chromosomes. This is we haven't looked at this yet, but I think this would be really interesting to look at, and also how that affects evolution.
Yeah, yeah. That's another really big, like, once you start exploring through this way that you have now to explore, it's almost like these other big, huge rooms of research
that's right.
loom that you could go into and
Yeah.
begin exploring. Like I said, once you realize there is a question that can be asked and then found the answers to, it's a whole new. Yeah.
Absolutely, and I think I'm not a virologist, but usually they say that the goal of the virus, especially like a retrovirus, is to kind of establish equilibrium with the host over in evolution, so
Right.
that it just stays around, it doesn't kill you. You benefit the virus, maybe the virus benefits you somehow, but in the long term, maybe the virus just wants to get along. Who knows?
Well, right. We all right. Yeah, if it does way too good of a job of killing its host, then it's
Out of
not
luck,
doing
right?
so
Yeah.
well.
Yeah.
Yeah, right. It shouldn't ought to do that.
Yeah.
Let's see. Ben again asks, and Ben, it's good to see you. Still not clear, but have the impression one goal of your research, I believe, is affecting cell differentiation and growth. And if so, do you feel applications look most favorable for fetal development or other all life stages, not just, embryo development?
Yeah, that's a great point, and I think these processes are important in a lot of other stages, just besides fetal development, for sure. They play big roles in the development of the embryo, but afterwards they're also important as well. So, for example, like Alzheimer's, a lot of the brain functions, they're detecting effects in adults. Also, I know for stress response, how you deal with stress, these effects also play a big role there, even in adults. So there are a lot of other areas that RNA is important in. So yeah, it is like, since the RNA science is relatively new, that means RNA drug development is also even kind of newer. So there's not, the vast majority of companies are doing protein-based drugs, and there's a couple that are doing RNA-based. If the Moderna vaccine works out, that's going to be huge for the RNA world, basically. Right. There may be a lot of other RNA companies coming out after that.
Yeah, that's fascinating. And he also adds another question at the end. And how would this differ from what CRISPR does?
Okay, CRISPR is CRISPR uses small RNAs, and its main purpose is to basically edit your DNA, the genome. And then in terms of these RNAs, these are just kind of RNAs flying around in the cell, but they actually do things. They like the brave heart is important for the whole heart developing. So the RNAs do lots of things in the cell, and then if there's an RNA that's, for example, causing a certain kind of cancer, if you could block that RNA, you might help with the cancer.
Yeah, yeah. And then if there were you could find that regenerative pathway. Yeah. It would be helpful to regenerate it no matter what age you're at.
Yeah,
if you need regeneration,
sure, absolutely.
Whether it's
that's
just
right.
to begin, like generation,
that's right.
or do something therapeutic.
Sure. That's
right.
Yeah.
What strikes you as sort of this big frontier that is being opened up by the kind of work that you're doing? What strikes you as, where it's going to go, or what you could see, what you might be able to envision 100 years from now?
Yeah, I mean, personally, I think like all the immediate issues are really thrilling to be working on, because it's so new and no one's seen what these look like. So in terms of just working out the details, it's exciting to see what they look like. But in terms of my own personal connection, I kind of moved. I started in astrophysics, actually, and then plasma, and then I moved to RNA research. And what drove me there was the origin of life problem. And many people think that in the beginning it was RNA, and then after that, or maybe some kind of modified version of RNA, and then after that came DNA and proteins. But since the RNA can carry information and fold up and do things too, it's a good candidate for the original source of life. And so therefore, I think that studying RNA, you can kind of study kind of the essence of life in terms of what was there in the very beginning. And so, for example, like the ribosome we work on, that's mostly made of RNA, and that's pretty much the oldest molecular artifact we have to connect us to when life actually began. And my hope is that that hopefully there's some sort of molecular fossils or remnants in these long RNAs of what happened a long time ago when it was only RNA. And then if we could try to understand the code, there's kind of a language both in DNA and RNA, and it's kind of like if you've ever thought of reverse engineering code, in computer code. So in computer code, you compile it into basically binary, ones and zeros, so it's not human readable. It's just this gobbledygook that you can't understand. But some people out there try to reverse engineer it by looking at little snippets of ones and zeros. Oh, this might be doing something or other. That's what we're kind of stuck with the human genome and also RNA. We just have this unintelligible code of A, G, C, and T. We don't know what it means, But the idea is maybe by looking at RNA, which hopefully was there from the beginning, these might be some of the key pieces of the code that we could try to understand and build up from there. So that's what I'm hoping to get in the long term out of looking at the structure of these RNAs. But,
I love that thought of molecular fossils.
Oh, right, yeah.
That's a great thing. Being a paleontologist,
Yeah,
it's like,
yeah.
It's in there.
Yeah.
That remnant, that original fingerprint of what it was.
Kind of like an RNA paleontologist, I guess.
Wow. Yeah, there you go. Oh, I like that. Very good. Let's see.
And you can imagine if you're a paleontologist and you come up with 98% of this stuff that's never been looked at before. It's like hitting a gold mine, like the pyramids or whatever. So here, suddenly the RNA people have found this huge amount of information to mine and try to learn from.
Right. So Ben comes back in again.
Okay.
Thank you, Ben. That brings up a chicken and egg question. Does mom require life, or does ma require life, or does life require ma?
RNA.
Is that what it is?
RNA.
I can't see that. Oh, geez, my vision. Thank you, Mel. Does RNA require life, or does life require RNA? Lowercase got me. Yeah.
Yeah, this is hitting on a very deep question, and this is deeper than most of the question. The normal deep question is chicken and egg. Did DNA, basically for DNA replication, you need proteins, but to make the proteins, you need DNA, so which came first? And then the answer to that was RNA, because it can presumably replicate itself and do things in the cell and do everything pretty much. But I think this person is asking something even deeper, which is, how did even the RNA world begin? And this, we don't really know, but the basic hypothesis is that, if you can, essentially the RNA, it's like a ticker tape string with a linear sequence of letters along it. And one of the sort of holy grails in this line of research now is to make an RNA that can duplicate itself.
Right.
And so basically you have to have the string long enough, and then it has to fold in just the right shape, so it turns into an RNA machine that actually can make copies of the RNA. And right now they've been able to duplicate the very short segments of RNA, but not at the entire, but not at, like if the little machine is, say, 1,000 bases long, they're able to duplicate maybe less than 100, but not the full 1,000.
Yeah.
But that would be basically the answer. So the idea is that once the RNA is long enough, it can fold into these complicated machines and start duplicating, and then that would be the origin of life.
And it's I'm thinking back to that, some of that original research back in, what was it probably the 60s,
Yeah.
with a soup
Yeah,
of carbon,
yeah.
hydrogen, oxygen,
right,
and
yeah.
nitrogen, and give it enough zaps.
Yeah, that's right, exactly,
Right.
yeah.
And you get strings put together.
That's right,
They
yeah.
form up. Well, so it's also sort of the old joke about, the infinite number of monkeys and the infinite
Yeah,
number
yeah.
of typewriters. Eventually, they'll write all the works of Shakespeare.
right, exactly, yeah.
Given in an infinity
Yeah,
of time.
this is exactly the idea. And so some people are trying to kind of prove this in the lab now. Can they make the RNA that can duplicate itself, I think.
Yeah.
And then, and one of the more controversial aspects, some people think RNA wasn't really stable enough, and maybe it had to be a variant of the RNA that's a little more stable, because I told you it cuts itself.
Yeah.
Everything's trying to cut it all the time. So some people are thinking it's a variant of the RNA that's much more stable that may have come first. But.
That's interesting. Something that had a little bit more self-preservation going on than RNA did. That
Yeah,
it
that's
wants
right.
to.
Yeah.
Yeah.
Right.
That's interesting. Chelsea asks, does your research focus on a specific type of RNA or all types of RNA at the same time?
Yeah, so I would say we tend to focus on very specific RNAs that are related to different diseases and that kind of thing. But most of the methodology is pretty broad and is applicable to almost any RNA, I'd say, yeah.
I mentioned early on from the info that you gave me that your research is partially funded from the National Institutes of Health.
That's right.
So
yeah.
they are obviously interested in therapy type.
Yeah, so we do a lot.
Applications.
We have a whole other line of research on these RNA switches, and these are a lot more direct than this, but they're in bacteria, but they actually fold up and directly turn on and off the gene. And those are interesting for antibiotics. The ribosome work we've done is the ribosome is mostly made out of RNA, and it's pretty much the main antibiotic target in U.S. hospitals. Most antibiotics you take, they work by stopping your ribosomes. So, and this is important for MRSA, drug-resistant bacteria, all this stuff like that. But that said, all the imaging methodology we have can be used on almost any RNA. And in our more theoretical work, we're interested in how magnesium. It turns out that this magnesium ion actually play a huge role in how RNA works, and we have a whole grant on how magnesium controls RNA. So, and that's that we just do one system on the RNA switch, but it's applicable to almost any RNA though.
So it's the way that the RNA works inside the cell. It isn't a component of the RNA.
It's not a component, but
Right.
it acts kind of like glue. And you're talking about evolution. It's interesting. So the RNA evolves its sequence so it can fold up, but it needs the magnesium around to catalyze that fold, kind of. So it kind of anticipates that there's going to be RNA, and then it uses the RNA to fold it up. So.
There you go.
Right.
Well, it was all in the same soup, and I guess it
Yeah, that's right.
kind of.
Right, yeah.
I'm just going to mention that we had the chance to work with you over the past few years
Yes.
and created an exhibit on
Yes,
the
that's
ribosome
right.
as a machine, as a
Right.
molecular
Yeah.
machine. And that exhibit, as you know, is here
That's
in
right.
the Bradbury, but it also travels around the country.
Yes.
And we've sent it around to many different places, and it is available to be borrowed by science museums and science centers anywhere for no cost. So if you happen to be watching us from not here in Los Alamos, but somewhere where you'd like to see that exhibit come to your home science center, science museum, get in touch with us. Get in touch with me or get in touch with Stacy Baker on our website and ask about the exhibit on the ribosome machine, which was just so much fun to put together. Yeah, great stuff. Let's see. I think we are at our time.
Okay.
Karissa, anything else that you'd like to add?
No, but keep learning about science and learning about RNA. So.
Yeah. Karissa, thank you so much. This was really fascinating. Really appreciate you being our participant here at Science on Tap. And thank you all to everyone out there who's participated. I look forward to you coming back next month and tell all your friends. This is a lot of fun. Thanks so much.